Hello, good morning, good afternoon, everyone. So I will continue from where I left and intend to finish this paper today. So I hope you remember that we are trying to read about a paper. We are trying to read a paper in generation of induced pluripotent stem cells, which should be transgene free, right? So the objective is to generate transgene free induced pluripotent stem cells. So as a part of tools in this, they have used, there are other methods, but then they were trying to use some things that are about the ideal iPSCs should be, one second for some reason. Sorry, one second, sorry. Yeah. Yes, the ideal iPSCs that this should not have any genetic changes. Okay, they should have appropriate or adequate amount of these pluripotency factors or Yamanaka factors expressed. Those are OCT4, SOX2, KLF4, and CMEC. And then reprogramming would have occurred. Because we are trying to use these for clinical purposes, one should ensure that there is no there are no any unintended genetic changes should not be there. We discussed about target site duplication for this. These authors have chosen piggybac transposons. Piggybac transposons are good transposons in contrast to other transposons. They do not leave a footprint of target site duplication. So I will quickly go through it. The transposons, the piggybac transposons, they make a cut here, double-stranded cut, and then they form a hairpin loop like this. They have an affinity for TTAA, okay? And they move by cut-paste mechanism. So they form a hairpin like this. In the integration site, as I said, they have affinity for, or they integrate in TTAA sites. So they make a cut somewhere here and here. So you will generate a fragment, two fragments such as this, and the other one is here. The transposon that is cleaved off from the donor site is next going to be cleaved here and here. And then you have these overhangs that are complementary to the target site, and they get integrated. Yes, it does seem like you have a target site duplication, as this was only one sequence in the beginning, TTAA, but now you have two sites, TTAA on left side of the transposon and TTAA on the right side of the transposon. But it's interesting to see what happens to the target site that donated the DNA, the transposon. This is the target site I'm trying to indicate here. So now they are cut, and you have two fragments like this, which are complementary. This overhang, the overhangs of these donor site fragments are actually complementary. So they just go and they base pair, and then they are sealed up by repair mechanisms. So at the end, they are not leaving any footprint of the transposon excision, right? This is in contrast to many other transposons that we know of. So eukaryotic, we discussed about eukaryotic mRNAs. They are monocistronic, and we should be able to generate 4 different proteins for a smart way of thing. For that, the idea, we also discussed about self-cleaving peptides, which can you can put, this should be the start codon, and this will be the stop codon. And a whole peptide, if this is the gene, a whole peptide like this will be generated with this start amino acid, methionine, and then you will have, and in between you have, and so on. We should have taken into account that the self-cleaving peptide should not have interfered in the function of the protein. Say P2A should not interfere in the function of this part of the protein, right? So these people thinking, or smart way of using this, that is 5' side of the piggyback vector, and it goes all the way. All of this is the transposon vector. Here you have the 3' side of the piggyback vector. Very small fragment of this is for manipulation within E. coli. Okay. So they have a CAG, a constitutive strong promoter, and then they have OCT4 reading frame. Then they have a self-cleaving peptide, SOX2 self-cleaving peptide, KLF4 and a self-cleaving peptide, and CMYC. And at the end, they also added a polyadenylation tail. I have given you 2 assignments, I think assignment 5 and assignment 6. In each of those, you will choose identify papers from the respective journals that are assigned to you, and find and describe how they have constructed the recombinant DNA in there. Okay, that will give you a way of understanding the literature and trying to figure out how they have done. It is going to be a good exercise for your understanding. And once you finish those exercises, in the future, you will be able to read any of the papers with much more ease. And this is followed by another promoter, and then puromycin resistance and thymidine kinase, that is for positive selection and negative selection. That we have learned in transgenic animals when we were learning that. So we will try to do that again a little bit today. So the whole goal of this is to, the scheme actually is something like this. You take somatic cells, you give in this the vector with the piggyback vector transposon, and because of the expression from the promoter, you will have the production of all the 4. I mean, they will actually be produced something like this, and then they will, because they have added the self-cleaving peptide, they would be cleaved off in these places like this, and you would end up with the 4 different Yamanaka factors, that is OCT4, KLF4, SOX2, and CMYC. Okay, so you'll have a strong expression of these, and only some of the cells would have taken them, and then you should eliminate those cells that have not that have not got transfected. So in that case, you can use the transposon. You can use the positive selection and negative selection there. But anyway, even if you're not doing that, you can figure it out. We'll come to that in a short while. The selection we will read in a different way. So, and then from those, you will be able to generate, you will find some of them have changed to pluripotency. And then we have to analyze how many copies are there and where did they integrate, because that is important, because we, at the end, we want them to be transgene free, right? If we don't know where it got integrated, we don't know where to check if there is transgene is present or not. And we should know, say the transposon has got incorporated into one location, say this one, okay? If we don't keep track of where the integration happened, and say we are just basing on selection, like this case, we don't know if there is any mutation that may have occurred because of the transposition or excision of the transposon. So it's important to keep analysis of the how many copy numbers are there and where exactly they have got, the transposon has got integrated. And once you have analyzed those, you will pick the ones that are good for your analysis, and then you will now try to select for removal of transposon. Um, and then you will use negative selection there, so that you will figure out those that have got integration free induced float, those cells that are integration free are selected and again analyzed for these sequences in the location like I was describing. Is there any mutation? Is everything cleared off? We just cannot base only on the selection. Okay. So this is what is going to happen. The construction is like this. They have made two different constructs. One of it has Lin28, another protein, another regulator of the development, and one without it. Okay, so there is no big deal in that. They will say OSKM, L is for Lin28. If they did not use incorporate Lin28, then it would be OSKM only. Okay, the rest of the things would remain almost same. So in between these code reading frames for each of them, they have introduced, this is translational fusion, right? You should also get used to the terminology so that you'll be able to follow the literature or any discourse on recombinant DNA technology much more easily. So these are all these the reading frames for these particular proteins are translationally fused, and also in between them they have added the self-cleaving regions for self-cleaving peptides. So upon translation, it is mRNA is produced. It is monocistronic. Why is it monocistronic? There is one open reading frame, right? But of course, we have there is no stop codon within this location. There is no stop codon. There will be only a stop codon somewhere here. Okay. So after translation, it would get cleaved, and it will produce OCT4, SOX2, KLF4, CMYC, and Lin28. Now we are going into results. When you are supposed to present results in your assignments, I would want you to have a clear look at. Don't be trivial about it. Try to have a very good insights, in the sense, know what is the technique. That first thing we should know is why are they doing it, right? After that, what are they doing, and then what are they saying about it. That is how you can cut it down, and you will find it much easier. So here is the one vector without Lin28. The other vector is with Lin28, right? So I want you to also look carefully into these individual. So this is the purified. The first one is about here. This is done by Western blotting. So in your presentation, you should be able to present about the principle of Western blotting before you go there, but not too much in detail. But the critical point should be done in terms of the perspective of this particular experiment. So you should say something like they have used primary bodies against the primary antibodies against OCT4, and then you should tell what secondary antibody and how they visualized it. Those kinds of details will give a good impression. So the individual one is something like they purified the protein or expressed it somewhere else, and this is not isolated from the vector based ones. They just isolated from outside. Okay. It is also important to have the legend in your PPT, so that it will be clear for me as well to see what you're saying is right or wrong. Individual is the positive control. Okay. And they are unlinked factor expression vectors. In the sense, you take a cell and you must have had just one expression vector and only OCT4. Okay. And the negative control here, the minus that is present, that is just negative control transfection using EGFP expression. Say, for example, if this is a somatic cell or differentiated cell, completely differentiated cell, I am trying to transfect the vector and I'm going to try to see if this is going to be expressed or not. And hopefully there is expression of OCT4. Okay, say for example. You should also remember that this cell also has OCT4 gene, isn't it? Then how do you know if this if the OCT4 that is present in the cell is secreted, or I mean is expressed from the transfected DNA or from the genome? We should be able to figure that out as well. So if you're doing Western blot, and there are 2 points. First thing is there is an action of transfection. Then the second thing is you put in the OCT4. Okay. If I didn't mention, OCT4 can, it has a positive feedback on its own transcriptional unit as well. So OCT4 is a positive regulator of OCT4. OCT4 can produce, it will produce OCT4, which can influence the or increase the transcription of OCT4. It's a kind of forward loop. So what we are trying to do in this particular experiment here is, or trying to see, is if OCT4 is produced or not. So here we are going to see, first thing is individual one. We just put only OCT4. Those others put only the, in a vector, only OCT4, nothing else. So you don't need to add any self-cleaving peptide and so on. So they transfected. Into the negative control, they just transfected, but not OCT4. They transfected EGFP, expression vector. Because we want to rule out that the action of transfection itself is causing the increase in OCT4. Okay. Well, let's not complicate it, and just we will go as it is. Now they have performed Western blotting using either OCT4 antibodies. In this blot, they have done SOX2, KLF, CMYC, Lin28, and beta-actin is to determine, or kind of, it actually acts as a control for the quantity of protein one has added. Okay. Say if I load less amount of protein, total protein sample here in the first lane, in the second lane, I add little more than first lane, and in the third lane, if I add even more, and in fourth lane, I did not add any protein sample, say for example. Okay. If I add more sample, it will give more signal, right? Typically, then I cannot claim that in those cells there is high expression of OCT4. I can only say that if I have added equal amount of protein sample in each of these lanes, say I used 10 micrograms in each of all the lanes, and then I see that here it is more and here it is than the control, that would mean that in that case the cells have higher expression. Okay, it is always a relative count. How do you know if it is same amount or not? And one of the nice ways is to use beta-actin. It will tell us that we added same amount of, it is kind of normalizing for the amount of sample that we are adding, which would also imply that it is like taking protein from normal number of cells also, kind of thing. So beta-actin, if you see any of the Western blots, typically they use beta-actin, is because beta-actin is thought to be stable amount, the quantity of beta actin is supposedly stable for a type of for a cell type. Okay, I hope you remember, you know, actin filaments, it's a part of the cytoskeleton and so on. Okay, so let's just now get into the results. So in this case, you see a band because you have only OCT4, and here you have in OKSM, you have the signal is there. In OKSML also you have signal. There is a difference between OKSM and OKSML, comparing this with this one, the second one, OKSML. Somehow presence of Lin28 is increasing the rate of expression of OCT4, and probably also increases the rate at which pluripotency is induced. The same is the case with SOX2, and you can also see it for KLF4 and for CMYC as well. What you and also in for Lin28. In Lin28, here you do not have a band because it is not included in the vector. It is just OKSM. There is no L. That's why there is no signal in this case. Okay, so let me again try to add one more layer. This is how you are supposed to critically look at the results. Okay, once you become good with that, you will your ability to design experiments as well as interpret the results will become way too high, and that is part of research aptitude. It is the same thing. This critical thinking is important for your CSAR part C, and that is why I would want that you try to practice when you're presenting in your assignments. Try to practice this. If there is something wrong, it is still all right, but it is a good idea that you find a study partner or partners and then try to discuss with them before you start presenting. Okay, it's always important to be evaluated so that you can get some backing, I mean, to make it error proof. So there is one question, however. In this case, the normal one is here and the other ones are little bit higher, right? The individual one again in SOX2 is also here, and from OKSM or OKSML vectors, the band is little higher, indicating that there is a small increase in size. The same is the case here too. Here you do not, these two are kind of same, but this one seems to be little higher in CMYC. And in here, there is no issue. They both, in Lin28, they both are almost same. Why is that? That is one another important question. It is because you can, the answer is here. If you look at the constructs, this would have been cut here, isn't it? That means OCT4 is actually till here, right? But it is now coming along with the T2A peptide. And that is why it is now increased in molecular weight, and therefore it will travel, move slower than the normal one. And that's why you see that these two bands are little higher, because they are coming with the extra weight of T2A peptide. Right? You can see it here, isn't it? OCT4, SOX2, NANOG, they SOX2, KLF, and CMYC, they have this extra piece of the self-cleaving peptide, and Lin28 or whichever is the last one does not have. So try to see if you find that that you can observe that kind of thing here or not. SOX2 is also similar here. It has a burden. That's why, I mean, little bit of peptide. That's why it travels slower. You can also identify the same thing as in KLF4. In CMYC, in OKSM, that is the last reading or protein gene that is added. In CMYC, it doesn't have any addition of self-cleaving peptide. So these two are kind of traveling almost the same, almost. Whereas in OKSML, the CMYC has T2A peptide. So it has increased molecular weight, and that is why it is traveling a little slower. Okay. Lin-28, it is not tagged with or fused with any of the self-cleaving peptides, so it will show as the same. Okay. That is how you can look at it much more critically, and that will give you a lot more insights. I hope you understood that. So the next part, so here they have introduced, and now they have proved that the OCT4 or the OKSM factors are induced, and that is how they have purified the, or if they were able to isolate some strains that are that have transformed. I mean, I shouldn't use the word transform. That are reprogrammed to pluripotency. So we have certain situations that we need to also discuss about the selection. We have taken, we have done transfection, and there are several possibilities that it might, we are transfecting, adding it, and then we are trying to remove it, right? To the cell, we are adding at one point, transfection. After induction of pluripotency, we are trying to excise. The excised one may go and integrate somewhere else again, isn't it? Reintegration is a possibility, and we need to take that into account. So puromycin resistant ones are the ones that are got the ones that have got transfected. So when you take several cells and you add the vector, and some of them will be will get the some of them will get the DNA, some of them will not get. So when you grow it in the presence of puromycin, all these cells will die because they don't have any resistance gene against puromycin. Okay. So the cells that are transfectants are puromycin resistant. That is positive selection. We are selecting transfectants from that of non-transfectants. Then upon culturing for a while, they will become induced pluripotent. Then we now have to remove it. So we will have transposase transfection. That means because we now want the transposon to get excised from the DNA. So let me put it here. Assume that this is the DNA, okay, and then we have a transposon integrated. This transposon to be integrated, you need a protein, that is the enzyme, that is the transposase. So you will the transfectants with a transposase after induction of pluripotency, not before that. So those that get, what will transposase do? It will cut this DNA and it will remove it. There are two possibilities. Either it might get or it may go and get reintegrated somewhere else. The one that I want is the degraded one. I do not want any reintegration, right? It is only adding complication to it. I want to get rid of the transgene. So there are several possibilities. Excision happened and reintegration happened or not. These are the events possible. And if excision did not happen and reintegration did not happen, the cells will remain puromycin resistant and FIAU sensitive. It is almost same as gancyclovir. I hope you remember negative selection. If thymidine kinase is present, thymidine kinase, gancyclovir will be made into a toxic compound in the presence of thymidine kinase, HSV. So I hope you remember that in the on the vector we also have a thymidine kinase included in it. So the cells, if you put in the presence of ganciclovir, they will die because thymidine kinase will convert it into a toxic compound. So FIAU sensitive. That means if it is puromycin resistant and if excision did not happen and integration did not happen, then they would be puromycin resistant as well as FIAU sensitive. If excision happened and also reintegration happened, that means it got cut out of here and went and got integrated somewhere else, those cells are also puromycin resistant and FIAU sensitive, right? Because they have puromycin gene as well as thymidine kinase gene. If excision happened but integration did not happen, then they would be puromycin sensitive and FIAU resistant. So these are the cells that we are actually looking for. So once we have integrated the OKSM factors, we got the puromycin resistant, we induced pluripotent stem cells, then we put in transposase, and there are three possibilities that transposase may not have recognized yet, or in some cells it might not have removed. Those will be resistant, puromycin resistant, FIAU sensitive. In some cases, the excision may have happened, but also reintegration might have happened. In that case, they would be puromycin resistant and FIAU sensitive. Those cells in which the excision of the transposon happened and reintegration did not happen, those will become puromycin sensitive and FIAU resistant. Those are the cells we are going to work with. Okay. And then they have to they proved several ways using Southern blot and other things, whether it is multi-copy integration or single copy integration when they first did that. So, I mean, they have identified several cells, several lines, iPSC. They're called, first one is, I mean, they might have identified several of those transfectants which have lost the transgene, sorry, I will get back one step. I'll take a step back. Normal cell, somatic cell, or differentiated cell made into iPSCs using Piggybac vector, okay, or OKSM. Then they have to now remove the transgene, right? Removal or excision. So they have got several of these, and they've named, they gave a name, something like IPS25 or IPS28 and so on. Induced pluripotent stem cell, you can say. So they selected a strain called IPS25 and IPS28. And in that, they have excised the transposon, and then they got many colonies again, and then they named them IPS25 delta 1 or IPS25 delta 2, like that. Delta 1, delta 2. That means this is the first cell one in which is FIAU resistant. That means transgene has been removed and it did not get integrated anywhere else. So here you can see that IPS25 has two bands, which is indicating that there might be two copies integrated into IPS25. And when they did they included IPS delta 1 and delta 2 and also delta 4. You don't see any bands. This is done by Southern blotting. Okay. So here these four are from one cell. This is IPS pluripotent, and these are the derivatives of IPS25 in which the excision happened and reintegration did not happen. Similarly, they have done for IPS28 and their derivatives, something like this. These are the same IPS25 from delta 11 and so on. You can see that they are number of additional cells. Here is IPS 216, and then you have the places. This is to show that these, you should understand the selection of samples here would have been those that are giving positive results. Means they might have tried IPS, say, 10, and probably that did not get integration properly. Okay, that may have problems with the growth, that may have removal of the or the reintegration may have happened. Many things could have been there. This gel, I think they have done several gels. For the presentation in the paper, they are doing a final gel in which they can clearly establish that iPSC 25 had the transgene here, and in these cell lines, IPS 25 delta 1, delta 2, and delta 4, the transgene has been lost. Isn't it? That's fantastic way of showing things. Yeah, so I will just move on. I'm running short of time, I guess. So here is, they're proving that using sequencing. IPS 28 delta 6 and 28. So they have shown that in the region where this is the actual site, this must have been the transposon where it got integrated. And once they have sequenced it, they have the sequence upon excision of the transposon, it again reverted to its original sequence, TTAA. Compare it with this one, right? The same is the case with the other ones as well. TTAA, integration, and upon excision, you again have TTAA. So that is perfectly excised without leaving any footprint. And that's the important part of that. So I hope you remember, in this case, you would have to explain Sanger's method and automated Sanger's method. So I will talk about, I'm not going to talk about karyotyping. I would rather stick to the molecular techniques that we have. In B is using RT-PCR. Here it is not real-time. It's not real-time PCR. It is reverse transcriptase PCR. Their objective is to show whether the transcriptional factors are expressed or not. They don't have to quantify it. And typically, if it were a RT-PCR, the result will be at the most in terms of CT values, which should be in graph-like things. Usually, they just give the number instead of the graph like this. For reverse transcriptase PCR, they would have shown the gel like this. That means they have taken mRNA, they have performed reverse transcription, and upon that, they have used gene-specific PCR, and then they would have loaded on the gel, and then they would show it. Okay, so here it is for OCT4 in different cell lines. This is the differentiated cell, mouse embryonic fibroblast, and here you have embryonic stem cell line. The third line, the first one is just using water. Second is somatic cell or differentiated cell. The third is real embryonic stem cells isolated from some embryos. And here we have iPSC. Sorry, I will use this. Here you have iPSC 25 delta 1, here delta 2, 28 delta 6, and so on. What you'll notice is this is to show that there are several proteins that are now expressed. Here what we are seeing is at the level of transcription. Okay. So OCT4 mRNA is equal in concentration with what one would identify from embryonic stem cell, and it is not present in mouse embryonic fibroblasts. SOX2 is also present in all the cell lines, artificially that is induced pluripotent cell lines. NANOG so is with other transcriptional regulators, including DNMT and so on. There is c-MYC probably is expressed in mouse fibroblasts, embryonic fibroblasts. But then in the others, in the real embryonic stem cell, the concentration is little less, and that is the same you will find in the other ones as well. So that is how you try to understand what the gel actually says. The objective of that particular experiment is to show that important transcriptional regulators and chromatin remodelers, or those enzymes involved in epigenetics, are expressed, and their expression level is equal to that of the ones we would expect in embryonic stem cells. And because for that reason they have used the appropriate controls in that way. Okay, here is one ips 28 delta 8 is not having enough of the some of the proteins are missing, they're not enough in concentration. Probably they would have eliminated ips 28 delta 8 from there to propose it, as it might not be ideal for use in clinical purposes. The second one is about, I will quickly finish this. The fourth D is something that we need to see. I think it is about bisulfide sequence. I think you were asked to present it as a part of one of the assignments. I hope you all remember that. So it is simple bisulfide sequencing. Okay, I will come to, okay, I'll just quickly go here. So bisulfide sequencing is to figure out if DNA is methylated or not. More specifically, if cytosine is methylated or not in a particular place, such as this one. Okay, so allele 1 and allele 2. Assume that in one case you have a methylated cytosine in this location, whereas in allele 2, there is no methylation in this location. So we treat it with bisulfite, then alkylation, and then spontaneous, and then you denature it. And then you can do PCR based on using primers for that particular location, and the sequencing. I'll just go to the previous one. So after bisulfide conversion, what would happen is all cytosines will become into thymines, and all methylated cytosines will remain as cytosines. Okay, so if you see here, we will just follow the top strand. There is a C here and a methylated cytosine. A cytosine, second base, and the third base is a methylated cytosine. 4, 5, 6, 7. Seventh cytosine is also methylated, and 10th cytosine is unmethylated. Okay, M is for methylation. Upon bisulfide conversion, what happens is all cytosines will become uracils. Okay, they'll become uracil. So the unmethylated one is in second position and also in the 10th position. I'm only talking about the top strand, this side. And then when we do PCR amplification, if you have uracil, it will, so 1 second, sorry. I'll just try to put it a little more better. If there was cytosine, and when you're doing PCR, it would have base paired with, it would have resulted in incorporation of guanine in the complementary strand. But if this cytosine has changed to uracil, it would result in the incorporation of adenine instead of guanine. Okay, so that is how you can figure out if something is methylated, if cytosine is methylated or unmethylated. It's the easy way to differentiate. You can look at this sequence here. All the unmethylated ones have resulted in the end. When you're doing the PCR, you will see that it has become, unmethylated ones will show up as thymines instead of cytosines, whereas the methylated ones will remain as cytosines. Okay, so because if you convert U to A here, I'm looking here, and then again you do PCR, the lower strand would involve the incorporation of thymine instead of thymine in that location by base pairing. Okay, so the others have used bisulfide sequencing to show that even the epigenetics is the epigenetic reprogramming has happened. How would they show that? They would show that in OCT4, usually DNA methylation. I think I've described this in one place. CPG islands, the cytosine is usually the one that is methylated, and methylation of cytosine is usually results in repression or silencing. So genes are not expressed. If you look at this differentiated cell fibroblasts, you notice that in the upstream or the promoter regions of OCT4, you see a large number of you see the black dots indicates DNA methylation. You see that a lot of cytosins are methylated. The same is the case with NANOC, which is not included, but it's also one of the important regulators in pluripotency. If you see the same type of result for embryonic stem cells, you do not find, they largely remain unmethylated. The cytosins upstream of OCT4 in embryonic stem cells would remain unmethylated. That means C means unmethylated is mostly associated with expression. If it is the opposite of methylation, methylation is meaning to repression. Not always the case, but DNA methylation at cytosine in CPG islands is mostly involved in repression, but it's not essentially the same case. It depends on other factors too. Other factors are location of CPG island and whether it is in the location of where repressor would bind or activator would bind and so on. But typically, it would mean methylation of DNA is repression of expression. So these are the two induced transgene-free induced pluripotent stem cells the authors have come up with. And what they show very nicely is that the methylation is absent. You don't see methylation as much as you see in the differentiated cells, right? The same is the case with IPS28 delta 6. Of course, there are some differences with NANOC and other things, but they're close to getting induced pluripotent stem cells which is transgene free, and that is a good enough success, I think. Okay. There are many other results. All of them will be revolving around the same thing. I have to discuss about this one. I'll do it in the next class about chimeras and so on. How to show if the generated induced pluripotent stem cells are actually really contributing, are they functional or not. We will do that in the next class. Okay. Take care. I'll see you in the next class.